Hello, my name is Ali. Welcome to a short advanced tutorial about 3D flexible refinement in CryoSPARC version 4.1 beta. This tutorial covers mesh generation. Please do read the corresponding section in the tutorial on the CryoSPARC guide as well. As we saw in the first 3D Flex tutorial, there are 5 job types that make up the 3D Flex workflow, and the 3D Flex mesh prep job is used to create a tetrahedral mesh, which the 3D Flex model uses to represent deformations of the canonical density under that mesh. Here's an example of a tetrahedral mesh for the COVID spike protein. The 3D Flex model can move these mesh vertices around and thereby deform the mesh elements, or tetrahedral cells, or in shorthand tetra cells, and the density within each tetra cell is deformed correspondingly as the vertices are moved around. The use of a mesh is an important part of the 3D Flex model that's designed to provide strong regularization of the deformations that the model can learn. Without the mesh, given the noise in the single particle data, it's very difficult to avoid overfitting in a high-capacity model like 3D Flex. The model would just learn very noisy and spurious motion rather than focusing on the deformation that's actually happening to the protein itself. As we saw in the longer tutorial, by default, the CryoSPARC 3D Flex mesh prep job creates a single continuous mesh, as shown here in the blue, that spans the solvent mask that surrounds the canonical density of our protein. The solvent mask is also created during the mesh prep job based on a consensus reconstruction that's input to that mesh preparation job. The job, along with creating the mesh in terms of its vertices and cells, also assigns rigidity weights to each cell of the mesh so that some cells are considered by the model to be more rigid than others. This is useful for preserving geometry of the dense, rigid regions in the core of a protein, but also allowing empty space around the protein to deform as needed so that the motion model can move around parts of the protein that should move. By default, this mesh, as I mentioned, is fully continuous and covers the entire object. And that default mesh generation does work well in most cases, and you should always use it as a first cut to see what kinds of different motion and conformations the 3D Flex model is detecting. However, 3D Flex does support any mesh topology or any mesh construction that you might want to use. A totally custom mesh with potentially variable sized elements so that they're not all the same size, even vertices that are not uniformly spaced, or maybe even a mesh that might be based on some prior information or knowledge you might have about a target molecule could be used. This is an area that we hope to explore further in the future and find out better ways to represent deformations and construct useful meshes. In CryoSPARC 4.1, using the CryoSPARC tools system, you can actually make a completely custom mesh and input it into CryoSPARC for use in 3D Flex. Some of this is covered in an example notebook in the CryoSPARC tools documentation. With the 3D Flex mesh prep job, we have actually created a method that makes it easy to construct meshes with a custom topology and connections between different regions of a protein using a coarse segmentation of the consensus density. Before you start training 3D Flex, you can create a mesh in this manner, and that's the topic of today's tutorial. We use this method, for example, in some of the newer results that are on the tutorial in the CryoSPARC guide, and those will also be in a paper that's coming soon. And one example of that is the spike protein of the COVID spike protein. The spike protein has a base region, this conical region at the bottom, and it also has 3 N-terminal domains and 3 receptor binding domains at the top. Those domains are all connected to the base, and they're all flexible. They are also very close together, and at the faces between the domains can move apart, closer together, farther apart, or in a shear-like motion relative to the other domains. And this is where the default continuous mesh that the 3D Flex mesh preparation job creates becomes a problem. Since the interfaces between different domains in the spike protein are quite small, it's very likely that across every boundary between 2 domains that might need to move apart or shear relative to each other, there's a tetracell that spans that interface. So a single tetracell within our mesh will contain density on both sides of the interface between 2 domains. When that happens, the 3D Flex model, because of the topology of the mesh that we provided, is not capable of separating those two domains in a clean manner, since it's only allowed to deform each tetracell by moving around its vertices. It can't, for example, cut a tetracell in half or make new tetracells within the mesh. Those are fixed from the beginning of training the model. So when we do have a fully continuous mesh, that can cause problems for these kinds of proteins in cases where we want to resolve more intricate motion, especially of domains or subregions or subunits that are very close together but move independently. And although none of them move rigidly necessarily, they still move in a way where each one moves separately from its neighbors. And that's the kind of motion that we want to capture in this case. So in order to fix this problem, we can actually adjust the mesh topology by having the mesh not enforce continuous deformation between the domains. Of course, now we need to instruct 3D Flex in the way of creating a custom mesh or a custom mesh topology as to which parts of the protein to allow to move separately and which parts should remain continuous and have their deformations remain continuous. The overall method for generating a custom mesh that allows for these kinds of discontinuous motion at certain boundaries is to create multiple submeshes and then join those together in such a way as to create a single final mesh, but that mesh is no longer completely continuous. The mesh still covers our entire protein, our entire solvent mask, but now has different topology where not all cells are connected to each other. The way this is done, the way that we've constructed the 3D Flex mesh preparation job to make it straightforward to construct a mesh of this kind of topology, is by using segmentation, the idea of segmentation. So on the left here in part A, we can see our typical default generated continuous mesh for COVID spike protein, the side view and the top view at the bottom. The idea of this technique is to input a coarse segmentation, and the segmentation will show how to create using UCSF Chimera's Seger tool, although CryoSPARC also allows input of an MRC file that defines segments, and you can create those in any way you like. Once we have some segments defined, and those definitions of segments and their boundaries only need to be very coarse, separating the boundaries, but don't actually need to be shaped based on our expected result or expected shape of the density, especially for parts that are flexible. Once that segmentation is input, the method that's in the mesh preparation job will create multiple submeshes, one for each segment, and then join them together. I'll explain that all in a bit more of a detailed process, and we'll go through a walkthrough of actually using this tool so it'll become more clear. But in terms of the actual steps that happen, we input into CryoSPARC a coarse segmentation, and that is expected to be created based on a consensus rigid reconstruction that you have already before you start using 3D Flex. So this is not based on the results of 3D Flex, and it's not based on what we expect to find, but just the density that we have to start with, our rigid reconstruction density. We can use that to define a coarse segmentation. We input that as well as what's called a connection tree, a list of pairs of regions that need to be fused together or joined together to create our final mesh. The way the technique works is that first each segment is expanded, meaning that every voxel within our solvent mask is assigned to be part of one of the segments, whichever segment it happens to be closer to, so that in total all of our segment boundaries cover the entire solvent mask. And then for each one of these regions, a submesh is generated. Those submeshes are actually subparts of the overall continuous mesh that would otherwise have been created. But this time, each of those submeshes only covers one region at a time. And those submeshes may actually overlap and have some cells and nodes that are in the same positions as each other. And that's where the next step comes in. Submeshes are fused based on the connection tree that we provide. So in the case of the spike protein, we would want to fuse the cone region, the base region at the bottom, to each of the RBDs and NTDs, but we wouldn't want to fuse any of the RBDs or NTDs to each other so that they can all stay separated, and 3D Flex will be allowed to move them in ways where they can shear or move further or closer apart, with the motion of one not necessarily influencing the motion or deformation of the other. And this is what allows 3D Flex to break the boundaries and have motion be discontinuous at those boundaries. And so finally, once those submeshes are fused together, every voxel in the solvent mask is now, when it's being deformed, it's deformed based on the positions of the tetrahedral cells and vertices that the 3D Flex model decides for that particular region that that voxel belongs to. And that's what allows, for example, a single tetracell that otherwise would have covered both a blue and pink region here is now separated so that there are two tetracells that overlap each other, but one only deforms the blue density and the other deforms the pink density. Together, all of this allows us to create a single mesh that covers our entire object, but allows 3D Flex to model deformations in a more sensible and intricate way. Digging a little deeper into the concept of the connection tree, I'll just show a quick diagram of what this means. Imagine we have a protein density where we'd like to cut up the protein into parts so that boundaries between domains that are close together but not actually touching are separated, and boundaries between elements or regions that are actually continuous density are preserved. In this case, what we would do is break up our density into these different regions using segmentation, and then define our connection tree. The idea of the connection tree is to take the overall density and start from a particular root node. That's just a region of the density that we start from at the top of the tree or the beginning of the tree. And then each other node is a child of that node or a child of subsequent children. So that in this case, we would start with number 437 as our root node. And these numbers are chosen because this is how they will appear when we're using the example with UCSF Chimera's Seger tool. These are region identifier numbers. So region 437 has two children, 443 and 439. Those are these two nodes, one here and one here. And then each of those also has subsequent children. So 443 is connected to 440, and then 439 to 442 and 438. This tree, first of all, must be a tree when we're defining connections, and that's a limitation of how the 3D Flex mesh generation code works. There can be only one parent per region, and you have to start with a single root node, and there can't be any cycles. So we can't have a loop of regions where we go in a chain and then the last one connects to the first again. So as long as our topology is a tree, everything will work fine, and 3D Flex will break up the density into these chunks and then create a submesh for each one and then fuse the submeshes together along the boundaries between elements that we say are fused. So 437 and 443 will have a fused boundary, meaning that density in between them will be continuous and deformations will also be continuous. So as mesh nodes are moved around in region 443, those will also affect the motion through the rigidity penalty and just the overall connections of the elements in the mesh, the motion of density in region 437. Likewise, between 443 and 440, that connection will be fused, but between 437 and 440, there won't be any connection. So even if there are mesh elements that span across this boundary, each one will be from one side or the other, and so it'll only affect density on one side or the other of this segmentation. So in order to actually define this connection tree, in CryoSPARC, the input is simply a string of numbers that it pairs in a list of this form, where we use the greater than symbol to specify that one region is connected to the next one. So we start with the root region and then connect it to one of its children. And one important thing is when we're listing out the connections that make up this tree, we should list them out one level at a time in increasing depth. So we start with the root node and its children, and then each of the children of those, and then further down into the tree. So 437 connects to 443, that's the first connection. 437 also connects to 439, and then so on as we go along the tree. So you can create trees of any structure that you'd like, as long as they're trees, and they can also have any depth. Although we have not experimented with extremely deep trees. Generally speaking, we want to break up our protein into the fewest number of regions necessary to achieve the desired boundaries that we'd like. So in the case of the spike protein, for example, here, our tree has depth only 1, since we have one root node, that's the cone, and 6 children of that root node, and that's the entire tree. To make all of this more concrete, let's look at an example with the COVID spike protein that we discussed so far. Jumping over to CryoSPARC, I have a workspace where I've already processed some of the spike protein data through standard processing and non-uniform refinement in this case, as well as some flex refinement jobs. You can see the flex data preparation job, a flex mesh preparation job, number 100, where I created a default continuous mesh, number 135, which is an example using the segmentation that we're going to discuss now, and etc. So what we can do to start off to create a custom segmentation and a custom mesh topology is look at our flex data prep job. One of its outputs is a consensus volume, and that's a volume that's already downsampled and cropped to the same box size as our training data for training 3D Flex. And this is a starting point that we can use to create our segmentation and then our custom mesh. Of course, as mentioned, you can also make a custom mesh in any way that you'd like and input it into CryoSPARC using CryoSPARC tools. So I've gone ahead and downloaded this density consensus volume from our mesh preparation job and opened it up in UCSF Chimera. As we can see in this density, there's a fair bit of detail, but obviously it's at a lower resolution since we're only training 3D Flex at a lower resolution in this case. So the first thing we can do in order to create our segmentation that we'll use to define the mesh is to filter this input volume. We can filter it so that, and I've chosen a reasonable filter size here, we can filter it so that the filtered version of the volume still has boundaries defined for the different segments that we want to separate, but is a little smoother and simpler to work with. As I mentioned, the exact position of the boundaries is not important, nor is the shape of the segmentation that we do. All we need to care about is essentially the places where we want to cut the mesh. Those boundaries should be in the right place, but there's a lot of latitude in specifying this, and there's no question of hardness or softness of a mask or any other consideration there. We just need to separate our boundaries cleanly. So this is a reasonable filter level to start with. And now using this density, we can use the segmentation tool from Chimera. This is a very handy tool. And if you're using this, please do cite the Seger tool. There's a publication there. So we can go ahead and select our Gaussian filtered density and segment it. That'll just take a moment. And we can see immediately that this density has been segmented into a few regions. Obviously not exactly the way that we want to cut up our density, but this is where the Seger tool becomes very useful in that we can go ahead and ungroup and regroup different segments to make our final segmentation that we'd like. So we'd like to separate each of the 3 RBD domains that are up here, as well as the 3 NTD domains that form the outer radius of the spike protein at the top, as well as our cone region at the bottom. We also have the spike protein also has a flexible tail at the bottom. And this is an interesting part since this is something that we actually want to ignore in our modeling. We don't want to spend model capacity to model the flexibility of this tail. We really want to focus on the RBD and NTD regions. So this is something that we can separate into a separate region. And I'll show later with the mesh preparation job, we can specify that this region should be modeled as being rigid. And that'll stop the 3D Flex model from learning deformations of that region. So that can be an interesting way to also separate parts of the structure that we care about and those that we don't. So now continuing with this example, I can go ahead and use the tools that are part of the Seger tool so that I can separate and resegment some of this density. So I'm going to go ahead and ungroup this region, ungroup this region, ungroup this region, and then I can select multiple parts that make up each of the domains that I care to keep together and group those. So now this yellow density is one RBD, this red part is another NTD. These two pieces make one of the down RBDs. This is another NTD, and these two regions make up another down RBD. So these 6 regions are the top parts of our protein, and I can hide those. And I can also take the rest of the density that remains, select all of it, and group it all together. So now what we're left with, if I show all of the regions, are 7 regions, as we hope to get. The first being the cone region at the bottom, and the other 6 being the NTDs and RBD domains. Now, our segmentation is finished. Of course, you can make this segmentation more precise or better and spend more time playing with it, but as I mentioned, the boundaries really do not need to be precise. They're just coarse boundaries that need to separate our domains, and that's all that's really of importance here. In order to use this file, we can go ahead and save the segmentation file from Chimera, and that'll save the file as a .seg file. And this is the file format that CryoSPARC is able to read in the mesh preparation job to use this segmentation. You can also optionally provide CryoSPARC with an MRC file where the segments are separated so that every voxel is labeled, every voxel has a value that's equal to the index of the segment that it belongs to. So solvent regions that are not part of any segment have a value of -1, and every other voxel is labeled with 0, 1, 2, 3, 4, 5, 6, etc. for the different segments that are part of the entity. So that way you can create your own segmentation in any other tool, and as long as you can output it in a format like that or use your own scripts and tools to generate that kind of MRC file, you can input that into the CryoSPARC mesh preparation job. So now that we have this segmentation done and we've saved that seg file, we can go ahead and look at the regions attribute table. And if you're using Chimera X, you may not be able to open the attribute table, but if you hover over a segment, you'll be able to see the segment numbers, which are what we care about. So in this attribute table, we can see each segment number or region identifier. These are the numbers at the beginning here, 295, 308, etc. And we need to note down these numbers. So here you can use pen and paper or whatever else you'd like to figure out which regions are which. So you can give each one a name, and then also figure out the connection tree at the same time. So in this case, we would need to note that segment 311 is the root of our tree, and the others are all children of 311. And from that, and using the description of the connection tree that I gave before, we can create our string that defines all the connections with using the greater than signs. And these are the two things, the seg file and the list of connections between regions that we'll need to input into CryoSPARC when we try to make our own custom mesh. So I'm going to go ahead and create a mesh preparation job. And here, the mesh preparation job takes in two inputs. The solvent mask is optional, and the consensus volume should be coming from our data prep job. So I'll go ahead and connect those. I won't connect a solvent mask since I'll use the parameters to create a solvent mask within the job. And I'll just go ahead and see what parameters we used in this previous run. So a mass threshold of 1 and a mass dilation factor, or rather distance of 4. And once those parameters are set, I can go ahead and now, rather than leaving the segmentation file path blank as we've done before, I can go ahead and navigate to and find our segment file. Along with that, we can provide the segment list as a segment connections input using the format that I described. And finally, we can specify the names or numbers of any segments that are to be modeled as rigid. So in the case that I described with the spike protein, it may be useful to separate the tail here into a separate segment and label it as rigid by providing that in this comma-separated list of region IDs. Once all of those inputs are provided, and I'm going to just show this job number 135 where this has already all been done. So we provided some parameters for solvent mass generation, and then provided a seg file as our input, and then a list of connections as I described. And then in this case I did separate the tail, and so I labeled that tail as a rigid segment. Once those are all provided, we can go ahead and run the job. The job starts out in the usual way with the consensus volume filtered, thresholded and dilated and padded to make our soft mask, and that defines our final hard solvent mask. And as a sanity check, our filtered consensus reconstruction with the mask applied. And then the basic tetrahedral mesh is generated and the segment file is loaded. And this is where things get more interesting. So we can see here that the segment file was loaded from the path that was provided. A bunch of regions were identified from that file, and we get a plot of what the segment file contains as the definition of the regions. And this is in the same format that I described. So background regions are -1, and then every other region just has a value that's equal to the index of its segment. So segment 0, which is segment number 216 from Chimera, is in dark blue, and then light blue is segment number 1, and so on. So we can see here that the tail region, as I mentioned, was separated from the rest of the protein. The segment connection tree is then parsed and displayed. So we can see in this case, all the segments are just joined to the same root, as mentioned, or rather the tail is a root, and then it's connected to the cone, and that's connected to all the other segments. And the next thing that happens is expansion of the segments. So each segment is expanded to fill up the solvent mask. Every voxel is added to the segment to which it's closest to. And this is again the reason that our segmentation does really not need to be very precise, or it doesn't really matter what the shape of the segments are, since they're going to be expanded in this way anyway, and it's only the boundary in between the segments that is maintained. So once those segments are expanded, then the next phase of the method goes ahead and splits up the original mesh, fuses connected segments, and reassigns voxels to the correct tetrahedral elements, and that gives us our final mesh, where every different part has a different submesh, and the submeshes that are intended to be fused are connected together. Finally, we have our rigidity weights, and here you'll see that this plot looks a bit different, and that's because the tail region was marked as being rigid, and the way that we do that with 3D Flex is to simply give it a very high relative rigidity penalty, by default 20, a value of 20. So this region is 20 times more rigid than the most rigid normal tetrahedral cell. And that's the end of the job. So now this job has outputted a 3D Flex mesh, and we can go ahead and connect that to a 3D Flex training job, as I've done here. And these jobs will now load up that tetrahedral mesh and commence training in the usual way, but this time using that new customized mesh topology. And that can yield some really interesting results, and we encourage you to try this out and see what you find. Of course, as I mentioned, you can also make your own mesh, you can adjust the rigidity weights of the mesh, and there's many other things to play around with, but the 3D Flex mesh prep job makes it easy to at least break up the density into parts or regions that should move independently from each other, or that the model should be allowed to move independently to each other. It's important to keep in mind that the 3D Flex model is always modeling non-rigid deformation. So by making these segmentations, we're not labeling the different regions that we identified as being rigid subparts. If you've used multi-body refinement or other tools, local refinement, for example, those tools assume that different segments that we're labeling using masks are each rigid, and there are only a few degrees of freedom. 3D Flex instead is still modeling a complete non-rigid deformation of the entire object. And it is able to model correlations or similar motions of different regions, even if they belong to different submeshes. It still has the full freedom and flexibility to do all of that. We're just, by creating this mesh topology, also giving it additional flexibility in moving adjacent regions in different directions, for example.